Hey guys, today we're going to get into heredity a bit more than we have been. So we're going to be looking at how alleles get passed from parents to offspring. In order to do this, we're going to be using Punnett squares. We're going to start by reviewing some of the smaller type Punnett squares that you would have done in grade 9, and then we're going to get into something called a dihybrid square, which involves a 4x4 grid instead. Okay, so this is the heredity and genetics lesson. We're going to be looking at the different generations that are involved, so like P1 generations as well as F1 and F2 generations. We're going to be looking at what Mendel contributed to genetics as well as his laws. We're going to go over some definitions about heredity like heterozygous, homozygous, dominant, recessive, genotype, and phenotype. And then we're going to be essentially using Punnett squares to figure out offspring phenotypes, parent phenotypes, genotype ratios, phenotype ratios, all that kind of stuff. So heredity is the transmission of biological traits from parents to offspring. So essentially how traits get passed between generations. Genetics is a branch of biology that studies how inheritance happens, and all biological traits are determined by genes. Again, genes are specific regions or segments of the DNA that code for specific proteins. Gregor Mendel is sometimes referred to as the father of genetics. He experimented with garden peas to see how physical traits move between multiple generations, and he kind of came up with our whole understanding of heredity. The reason Mendel chose pea plants is because they're easy to grow and pollinate, they grow really quickly, and they have a bunch of easily identifiable traits. And many of those traits come in two forms. So he was able to very strictly control the reproduction of the pea plants by actually like transferring the pollen from one plant to another. And then he chose 7 traits that were easy to identify and had 2 forms to see how they would get passed between generations. So these were the traits that he picked for his pea plants. So he would like look at how seeds shape or how pod color or that kind of stuff move between generations. Based on these studies, he came up with 3 laws. The first is the law of dominance, which says that in heterozygotes, one allele will always be dominant over the other, and the non-dominant allele will be concealed. So what that means is you always have 2 alleles for all of your traits. If you're heterozygous, you have a dominant and a recessive allele, and he is saying with the law of dominance that the dominant allele will show up, the non-dominant or the recessive allele will not show up. He also came up with the law of segregation, which says that, again, all individuals have 2 alleles for every gene, and when gametes are formed, those alleles will be separated into different gametes. Therefore, in the end, you always end up with 1 allele from your mom and 1 allele from your dad, 1 from the sperm and 1 from the egg. And then lastly, we have the law of independent assortment, that says that alleles will separate into those gametes at random because if you remember independent assortment, which happens during metaphase, is when the homologous pairs line up at random across the middle of the cell. So he was kind of the one that came up with the idea that that happens randomly. When we're talking about different generations, we often refer to as the parent generation as the P generation, and then their children are often called the F1 generation, and their grandchildren are called the F2 generation, and then it would keep going from there, F3, F4, etc. When experimenting with peas, Mendel would use what he called true breeding plants as the P generation, which are plants that always have the same physical traits every generation. So essentially they're homozygous for their trait. Okay, so I've already used some of these words, dominant and recessive. Most genes can either be dominant or recessive with a dominant allele. That means that if an individual has the dominant version of a gene, then that trait will always show up. We always represent dominant alleles with a capital letter, usually the first letter of whatever the trait is. So we would say a capital T for tallness, because in Mendel's P's, tall is dominant over short. The recessive allele is the genetic option that will only be expressed if the person only has recessive alleles. So if there is a dominant allele that person has, their recessive allele will not show up. And we represent these recessive alleles with a lowercase version of whatever the dominant allele was represented by. So shortness would be represented by a lowercase t, since tallness was represented by a capital T. And so everyone has 2 copies of every gene, and those 2 copies could be 2 of the same alleles or 2 different alleles. If both of your alleles are the same, then you are homozygous for that trait. And you could be either homozygous dominant, in which case you have both dominant alleles, or homozygous recessive, which means you have both recessive alleles. If both your alleles are different, then you are heterozygous. So you have one dominant and one recessive allele for that trait. And then genotype refers to the actual alleles that you have. So whether you have like the 2 big letters or a big and a small, or the 2 small ones. And then the phenotype is the actual physical trait that you end up with. Are you tall or are you short? Usually there are 3 genotypes and 2 phenotypes. And that is because both the homozygous dominant genotype and the heterozygous genotype both give you the same phenotype. So if we look at this example here, big T, and big T, little t, both result in a tall plant or tall person or whatever. Because as soon as you have a big T, that is the dominant allele, so it's going to show through. And then if the person or plant is little t, they will be short. It is important to note that the dominant trait therefore has 2 different possible genotypes. The recessive trait only has 1 possible genotype. And so usually what we're looking at when it comes to genotypes and phenotypes are actually genotype and phenotype ratios. So we're looking in a genotype ratio at what is the ratio between homozygous dominant, heterozygous, and homozygous recessive. What ratio between those is expected in the offspring of 2 parents. When we're looking at the phenotype ratio, we're looking at the ratios of the different traits, so the dominant trait versus the non-dominant trait. These ratios are usually written as like number, colon, number, colon, number. And then we can turn that into a probability that we write either as a fraction or a percentage. So when we're doing Punnett squares, this is how we figure out those probabilities. And they can be either monohybrid or dihybrid. If we're only looking at 1 trait at a time, we will use a monohybrid Punnett square. These would be the types that you did in grade 9, where it's like 4 boxes, like a 2x2 square. Dihybrid crosses or dihybrid squares look at 2 traits at the same time, and they will be a 4x4 square, so 16 boxes in total. Usually you're given the genotypes of the parents, and you use that to figure out the genotypes and phenotypes of the potential offspring. So you can work backwards from the genotypes and phenotypes of the offspring to determine the parent genotypes as well. So the normal steps that we use in Punnett squares is to figure out what the genotypes of the parents are if we're not told. Then we determine what the gametes of the parents are. Then we are going to write the gametes of the parent on the top and side of the Punnett square, and then we just fill in the square. So let's do an example of this together for one of the monohybrid squares, and then you guys can try the bottom two on your own. Okay, so in this first one, it says that roundness is dominant over wrinkles. So I'm going to say that a capital R means round and a lowercase r means wrinkled. Just to kind of figure out what my alleles are going to look like. So those are my two alleles, my two possibilities for the shape of the seed. And it said a wrinkled seed is crossed with a heterozygous seed. So my two parents, we'll say parent 1 is the one that has the wrinkled seed. Now, in order to be wrinkled, you must have 2 of the recessive alleles. If you had one of the dominant alleles, you would end up round. The only way to become wrinkled is to have both recessive alleles. And then parent 2 is heterozygous. So that means it's going to have a big R and a little r. Then to figure out what the gametes are going to be, in a monohybrid cross like this, we just separate the 2 alleles. So those are our 4 gametes. And then a monohybrid cross is just a 2 by 2 square. I'm going to put one parent down the side, and I put one allele kind of per box on the outside, and I put the other parent across the top, again, one allele per box. Doesn't matter which parent goes on the top and which one goes on the side. And then I'm just going to fill it in. So to fill this box in here, I just go to the top and the side, and whatever's there, I write them. We always write capital letters first, so I'm going to write big R, little r. And then I'll do the same here. I go to the top and the side to figure out what to write in the box. And we always write capitals first, so big R, little r. And then here, go to the side and the top, it's little r. Here, go to the side and the top, it's little r. Then it wants to write the genotype and phenotype ratios. So the genotype ratio is always written as homozygous dominant to heterozygous to homozygous recessive. And we just count how many of each of these there are. So we have no big R, we have 2 big R, and 2 little r. You could also put these in lowest terms and write that it is a 1-to-1 ratio between your heterozygous and your homozygous recessive. The phenotype ratio is always written as dominant trait, I spelled dominant wrong, to recessive. And so we go over to our grid here and we count how many of these 4 are going to result in a dominant trait. And the answer is 2 of the 4. Anytime we have a capital letter, it will be the dominant trait. So 1, 2 will be dominant, and the other 2 will be recessive. Again, we can put that in lowest terms as a 1-to-1 ratio. And then lastly, it says, what is the probability of a round seed being produced? Well, the round seed in this case is the dominant trait. So I could say that 2 out of the 4 are going to be round, which is a half. And I could also divide that to say 50%. So a half or 50% will be round. The other half is going to be wrinkled. Okay, so now try the other 2 on your own. Okay, so for number 2, we're told that yellow is dominant, so we'll represent that with a capital Y. Green is recessive, so it gets a lowercase y. And then we're told that both parents are heterozygous, meaning that both have a big Y and a little y. So I put the big Y and the little y on both sides, and then I fill in the grid. Make sure that when you're filling in the grid and writing out your alleles, that you make sure the uppercase and lowercase letters look different from each other. Try not to just write one as a smaller version. Try and make one like actually distinct. Like here, my uppercase has a straight tail and my lowercase has a curved tail. Make them distinctly different. When you guys start doing this on tests and stuff, I will give you the option of taking pictures of your work to submit to me. And because there's no diploma this year, I can give part marks, but I can only do that if I can tell your alleles apart. So make sure that you're doing that. Then for the genotype ratio, we have 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. And then for the phenotype ratio, the dominant trait, which is yellow, would have 1, 2, 3 versions. And the green would just come out once out of these 4 options, only when you have the little y, little y. And then lastly, what is the probability of a green offspring? Well, that is the 1 little y out of the 4 boxes, or 25%. Okay, number 3 is a little trickier because it's asking us to work backwards. So it says that purple flowers are dominant. Oh, that should say purple. Purple are dominant and white is recessive. Again, I have made my capital P have like a double line on it so I can tell it apart from my lowercase p. And then it says the parent one is white, which means it must be lowercase p, lowercase p. So I'll put that on the side of my Punnett square. It says that parent 2 is purple, which means that it definitely has a capital P, but we don't know if the second allele is a dominant or a recessive allele, because it would be purple either way, whether it's homozygous dominant or heterozygous. So I start off by filling in this first column here, just based on the information I know. And then I'm told in the question that half of the offspring are white, which means they must be little p, little p. And then I can work backwards and say, okay, well, one of these little p's came from the side, the other one must have come from the top. Therefore, the genotypes of the parent generation must be little p, and big P, little p. Okay, next up we have the dihybrid crosses, which is when you're trying to determine the likelihood of 2 different traits appearing in offspring at the same time. In this case, parents will have 2 genes each, which is 4 alleles. So when you're making their gametes, you can't just separate them anymore like we did with the monohybrid cross. Instead, you actually have to foil the 2 genes. And I'll show you what that looks like. Then you write the gametes on the outside of your 4x4 square, just like you did last time. And then you fill in the square. Always pick one of the traits that you're going to write first all of the time. And then we always write our dominant allele first as well. We're always going to assume that genes are sorted independently unless we're told otherwise, because that's the only way that dihybrid crosses work. So let's go through these 2 examples. Okay, so in example 1 here, it says that yellow and round are dominant to green and wrinkled. So we have yellow and round, which are capital Y, capital R. So then green and wrinkled our recessive traits must be lowercase y, lowercase r. It then says one parent is homozygous dominant and the other is homozygous recessive. So that means if parent 1 is homozygous dominant, they are all capital letters. And the other parent is homozygous recessive, so all lowercase letters, like so. Okay, then to find their gametes, we FOIL. So when you're FOILing, you start with the first letter and go to the first letter of the next gene. So that's going to be YR. Then I go back to that first letter and bring it to the second letter of the first gene. In this case, it's also YR, big Y, big R. Then we go to the second letter of the first gene and bring it to the first letter of the second gene. And then we go back to the second letter of the first gene, bring it to the second letter of the second gene, and we get our 4 gametes. Now, when you have a fully homozygous dominant or fully homozygous recessive parent, all 4 of their alleles will be the same, but that's not always going to be the case, because if you had a lowercase letter anywhere in here, then they wouldn't all be the same. So I'm going to do the same thing on this side, and I'll end up with, in this case, all 4 of the gametes being the same. Then this time, because we're dealing with 2 traits, I need to make a dihybrid cross. Which looks like this. And then I'm going to fill in the gametes, 1 down the side and 1 across the top. And then I'll fill in the square again. So I'm going to write my Y's first and my capital letters before my lowercase letters. So I'll write my capital Y and then my lowercase y, capital R, and lowercase r. And it's going to be the same the whole way across because the gametes are all the same. It's also going to be the same the whole way down because the gametes are all the same. And feel free to just write arrows to fill in things that are the same. You don't have to actually write it out every single time. In this case, it wants to know what are the chances of a yellow wrinkly seed. Well, in order to be yellow and wrinkly, you would need a capital Y for the yellow, and then it wouldn't matter what your other Y allele was. It wouldn't matter if that's big Y, or big Y, little y, because yellow is dominant. And in order to be wrinkled, you would have to be little r, because in order to get a recessive trait, you need both recessive alleles. So what are the chances of a yellow wrinkly seed? 0%. None of these are going to be wrinkly seeds. Okay, try number 2 on your own now and then come back to check your answers. Okay, so for question number 2, we're told that both parents are heterozygous, meaning that they have a capital and lowercase letter for both of their genes. When we FOIL this to get the gametes, you should have big Y, big R, big Y, little y, big R, and little y, little r as your 4 gametes. And then you put them on your Punnett square and fill it in. Again, you pick one of the letters to always go first. So with me, Y's always go first. And then if within the Y's or within the R's, you have a capital and a lowercase, the capital goes first. Then you were asked for the phenotype ratio. So to set up the phenotype ratio, you're looking at the 4 possible combinations of traits. The plant could be yellow and round, yellow and wrinkled, green and round, or green and wrinkled. And then in order to identify those, I kind of write the possible genotypes that I'm looking for on top of the phenotypes. So yellow and round is anything with a capital Y and a capital R. For yellow and wrinkled, I need a capital Y and 2 lowercase r's. For green and round, I need 2 lowercase y's and a capital R. And for green and wrinkled, I need all 4 of my alleles to be lowercase. What I do now, because I'm very much a visual learner, is I go through and label each one of these with a color, and then I color code my Punnett square. Okay, so I have color coded my 4 different possible phenotypes, and then I'm going to look in my Punnett square and say, okay, where do I have all lowercase letters? And that is right there. And then I'm going to look and say, okay, where do I have all lowercase y's and a capital R? Well, I have that here and here and here. And I think that's it. And then I will look for my capital Y with lowercase r's. So capital Y, lowercase r's are those 3. And then lastly, I'll look for anything that has a capital Y and a capital R. Which is all those ones. And then I'm just going to count them up. So I have 9 yellow dots, 3 pink, 3 green, and 1 orange. And this ratio here, 9, 3, 3, 1, is a special ratio because anytime both of your parents are heterozygous, you will always get a 9, 3, 3, 1 ratio, where you have your fully homozygous dominant as the 9, fully homozygous recessive as the 1, and your mixtures where 1 trait is dominant and 1 trait is recessive are the 3s in the middle. So that is a ratio that you can just remember. Then it says, what is the probability of a green round seed? Well, that would be 3 are green and round out of a possible 16. And if you divide that, you get about 19% as your probability of a green round seed. Okay, so that's it for our dihybrid crosses, though I will have more worksheet practice questions coming for you later. The last thing I want to talk about is linkage groups. So I said that in order to do these Punnett squares, we have to assume that our genes are separating independently, but that's only true if the genes are on different chromosomes. If genes are located on the same chromosomes, they're often not able to separate or assort independently because the entire chromosome is going to go one direction or the other. So instead, we call them linked. So those genes become a linked group, and they're usually inherited together. The closer together that genes are on a chromosome, the more likely it is that they will be inherited together. If they're further apart, there is a chance that they could get separated during crossing over when alleles and genes are swapped between chromosomes. But if they're really close together, it's likely that even crossing over wouldn't separate them. So if we look at that picture on the top, which is showing 2 different chromosomes, the flower color and the resistance to virus are fairly far apart. So they could get separated with crossing over, whereas plant color and leaf ridges are quite close together. They're probably not getting separated. So therefore, it's most likely that if you have 2 traits inherited together, it would be the plant color and the leaf ridges. We're going to talk more about this later on as well.